recording.
Hello, everyone. Thank you for joining me. This is Melania Figueroa from the CSIRO, and today I'll be talking to you about the role of somatic hybridization in the evolution of the Ug99 lineage of the wheat stem rust pathogen. And I'll mention that this work was recently published, and I'll take this opportunity to thank all the authors on the manuscript, but in particularly Fong and Narayan, who shared first authorship, and also senior authors Peter Dodds, Benjamin Swessinger, Brian Stephenson, and Zach Pretorius. And I will also like to thank the Two Blades and the USDA NIFA for support of this work. And also highlight here the role of women in science. And in particularly, I want to highlight the work of Yana, Eva, and Marisa, along with Fong. And I want to thank them so much for the work that they done through the years and that enable this work. Okay, so this piece of research really, this journey started with us attempting to separate haplotypes in rust fungi. And as you are aware, rust fungi have two independent nuclei, and each of these nuclei contributes to the infection process. So if we separate the information from each nuclei, we are well positioned to define genetic factors that control plant infection. As I'm showing here, this strain is heterozygous um, for an virulence effector locus and therefore unable to infect the plant. And here this strain is is homozygous for the virulence allele and therefore infects the plant. And another benefit of having this resource is that we are well positioned to understand virulence evolution. And as Peter mentioned earlier, we can track changes on a heterozygous strain, for instance, point mutations, insertions, or deletions. So really this work started by us selecting two strains, 21-0 and Ug99, and acquiring PacBio data for both. PG21-0 is a strain that is very predominant in Australia, and it came from South Africa in the 1950s. And we applied this assembler called Canoe. And as you can see here, the size of the assembly were actually twice the size of a haploid genome size, meaning that we were very successful in acquiring all the information for both nuclei. And so our next step was to assign those contigs of the assembly to the respective nuclei and therefore haplotypes. And we took here a synteny approach by really using a reference contig as bait and looking for contigs that share a number of genes in common, as I'm showing in this example. So we would end up with something that looks like this, an alignment of a large contig with 4 contigs or more that are matching, so representing the alternate haplotype. And this worked really well. And in some cases, we managed to reconstruct a whole chromosome. And one of those chromosomes um, was actually the chromosome that contains ABRSR50 and ABRSR35. And reconstructing both haplotypes. And one of the surprises is that one of these haplotypes in 21-0 was highly similar to that in UG99. So this really made us wonder if we were in presence of a situation like this, but the both strains share one chromosome, or actually it was telling us that in fact the two strains shared an entire haplotype. And to test that, we took a read mapping approach, a subtraction approach. We took Illumina reads from UG99 and mapped those to the genome reference of 21, and the reads that did not map were mapped back to UG99, and we did that vice versa. And what that would really tell us is if a contig was common to both strains, they could the coverage would drop to 0. And this approach worked really well because we were able to split the genome assembly in basically half. and it's provided us evidence that both 21 and UG99 were sharing one haplotype. And when we both we compare the context in haplotype A in 21.0 with the context in haplotype A of UG99, we found a high level of identity, about 99.6%, with a really very little structural variation between the two haplotypes. However, if we do the respective comparisons between haplotype A and B in 21, let's say, we see how that identity decrease and the structural variation increase. So this allows us to develop the hypothesis that UG99 is a somatic hybrid with one of the parents from the PGT21 lineage. And in this scenario, this member of the lineage donated the A genome after hybridizing with an unknown isolate that donated the C genome. And this became the progenitor of UG99. Alternatively, we could have something like meiosis and sexual recombination, in which case the A genome of UG99 was derived from the genetic recombination of the two haplotypes in this member of the PGT21 lineage. So to really examine the hypothesis, we looked at all contexts in 21 that display evidence of being chimeric, because those could be signs of recombination, but also they needed to be examined because in those cases recombination could also look like assembly errors. And in all the cases that we examined, 37 of them, we found evidence that in fact the assembler had lost phasing information, and those point of junctions in this context were actually collapsed regions displaying twice the coverage and also heterozygous SNPs. And we also validated that using Hi-C data, which would provide information about the linkages between these fragments. And that also told us that those chimeric contexts were indeed assembly errors. The Hi-C data also allow us to reach a chromosome level assembly of PGT21. And in this case, we were in a position to answer the question if chromosome reassortment could have had happened, and again, being related to sexual recombination. And after our analysis, we concluded that all the chromosomes in haplotype A were highly linked to other chromosomes assigned as haplotype A and not B, suggesting that the scenario we had is that it was this one that I'm suggesting here. So ruling out the potential for sexual recombination is supporting the evidence, supporting the hypothesis that UG99 was the product of hybridization of a member of the 21 lineage that donated the A genome in fusing with an unknown isolate that donated that C genome. And later that progenitor would have gained virulence through a mutation, perhaps in AVRSR31. So I will just finish up here by thanking the new group. I moved to Australia to the CSIRO over a little bit over a year. In November, I stepped as head of the serial ROS group. And so I really want to thank my new colleagues for welcoming and supporting me. Thank you very much, and I will be happy to join the discussion panel later for further discussion. Thank you.